I don't think that evolution is a teleological process. I don't think there is an end point, at least not one that's predetermined. I am a big believer in the role of contingency that Stephen Jay Gould argued fairly persuasively in favor of. He posited that if you could turn time back 500 million years and let it play forward again, would you get the same outcome? He thought that if you ran the tape forward 1,000 times, you'd get 1,000 different outcomes. That view is not universally accepted. There's a guy named Simon Conway Morris who very famously disagreed with Stephen Jay Gould about this. They had a series of exchanges in Natural History magazine. There are limits to what we can know. So we will probably never know how life evolved on Earth. The best we're ever going to be able to do is say what's plausible, not to say this is how it happened.
You are studying a fascinating question that everyone has a stake in. Why is it that there are creatures of many cells versus creatures of only one cell?
Exactly, right.
So do you have an answer for that right off the bat?
No, not one answer.
Okay.
There's probably a lot of answers for that because there have actually been a lot of independent events throughout the history of life on Earth where single-celled organisms have evolved into multicellular organisms.
And is that to say that it's an adaptive advantage to do so?
Probably, at least in some circumstances.
And that's what's so fascinating. It hasn't been across the board. We still have bacteria hanging around that are perfectly content to remain single-celled.
Exactly. I mean, the vast majority of life on Earth is single-celled, even today. So what that suggests to me, in combination with the fact that there have been lots and lots of independent origins of multicellularity, is that it probably is advantageous in some circumstances, not in others. And we should also allow for the possibility that not everything in evolution is about adaptation. Some things in evolution happen through neutral processes as well. So we can't really say for sure that any particular origin of multicellularity was driven by a particular selective pressure or by selection at all.
So you're speaking of accidental processes, or what's an example of these neutral processes?
So neutral theory is a big deal in evolution, and it's based on the fact that biological populations are all finite in size. So if you think about a sampling process where, say, I'm trying to think of a good example here. You've got a mixture of blue and red balls, and you're going to pick 10. And there's 60% red balls and 40% blue balls. So in theory, if you pick 10, you should get 6 and 4. But you're not going to get that every time, right? Sometimes you're going to get 7 and 3. Sometimes you might get 8 and 2. Sometimes you might get 10 and 0.
Mm-hmm.
And if you were picking an infinite number of balls, you would get a 60-40 ratio. But if you're picking a limited number, a finite number, a lot of times you're not going to get exactly that ratio. And so in finite populations, the frequencies of different gene variants can change from one generation to the next without any selection going on at all. And
just because it's random noise within the system as sort of reproduction happens.
And people are in the right place at the right time to reproduce. And
what's that?
Just creatures end up being in the right time at the right place to reproduce, and for whatever reason, exactly. There's luck and chaos, I guess, in nature as well.
Yeah, I said it in a much more long-winded way, but yes,
we try to break it down to, we're just trying to get the dumbed down version for ourselves, for folks who don't spend all their time looking at these matters.
And if you did that lots of times, say you picked 10 and you got 7 and 3 instead of 6 and 4, and then you multiplied each one of those by 10. So you had 700 and 300. You pick 10 again. Maybe this time you get 8 and 2. Maybe not. Maybe you get 6 and 4. Maybe you get 8 and 2. But you can actually lose a variant that way. If you do it enough times, you may find that eventually you don't have any blue ones at all. All you have is 10 red ones.
And the idea is that with enough repetitions, you'll lose some variants that are beneficial, but you'll acquire other ones that are brand new in the population, and they would sort of, with enough repetitions, emerge as being fixed in the population or present in the next generation.
Yeah, the only thing I would change about what you just said is that if it was, what we're talking about here is variants that don't affect fitness very much. So I wouldn't necessarily call them beneficial. We're talking about variants that don't have a big impact on whether you live or die, whether you reproduce or fail to reproduce. Those can get fixed or lost from populations just through neutral processes.
And how does this connect back to multicellularity?
I just wanted to make the point that for a particular origin of multicellularity, it's entirely possible that it happened through neutral processes. We shouldn't assume that every single origin of multicellularity was an adaptive or selective process.
Is it possible to say, though, that after it emerges and that it doesn't disappear, that it eventually proves benefit? Or is that something that, are there creatures that are multicellular with no apparent benefit whatsoever? They're just like walking around multicelled, but just, for no good reason.
Probably not. And the reason I say that is because we know there are costs to being multicellular. So, you need more food. You're more, let's see, what are the disadvantages to being multicellular? I guess you're going to reproduce more slowly. You need to eat more or photosynthesize more.
You also die in a different way, right?
For a lot of multicellular organisms, that's true.
Like it seems like there's more of a and this is we're getting metaphysical early on in the conversation, but it seems that multicellularity also, in addition to changing how you live, it also changes what death means. Because for a bacteria that undergoes binary fission, a yeast that undergoes binary fission, you don't have death really. You have this sort of dispersal into the population. But for a multicellular organism, there appears to be a moment where you can say that, okay, this is the end.
There's only one cell that doesn't die at the end or something like that.
Yeah.
There's a great science fiction story based on that premise. I think it's called, and I don't remember who wrote it. I believe it's called Mortal Gods. And the idea is that humans are the only species in the universe, the only intelligent species in the universe that dies, because everything else just divides the way you're talking about. In a lot of cases, though, it's actually more complicated than that. So a lot of times, even things that look like binary fission, or they are binary fission, they're not perfectly symmetrical. And if you really do track the individual cells, you will find that there is a lineage that dies. You can see that most clearly in budding yeast, where they don't split equally in two. They split very asymmetrically. They form a bud on the side. They're round, so they don't really have a side, but on their surface, they form a little bud. It grows a little bit, and then it breaks free. So you have a clear mother cell and a clear daughter cell, which you don't really have with E. coli, right?
Well, in some really old bacteria, the firmicutes are sporulating, so it's almost like they're budding off little seeds as well. It's kind of interesting.
Though I wonder if there's a difference there, though, like where the yeast, it seems like the mother cell can give birth to multiple daughters, like a single mother cell buds multiple times.
Multiple but limited.
So, and with the firmicutes, I think that it's a direct one-to-one relationship where one bacterium develops a spore, right?
But I guess it divides, and then the old parent dies off.
Yeah, that's true. So
it's just similar to the animals.
But so there's a limited number of times that the yeast can divide.
Right. And then it's basically dead. The mother cell basically dies after a certain number of divisions. So it's not entirely true for all unicellular organisms that they don't die in a similar way to what we're thinking of. But I always have thought that's a really fascinating, just kind of philosophical problem that, when an E. coli divides, you don't have a mother and a daughter. You've got two copies of what was there before.
And in the unicellular realm, is that more common or less common than having a sort of a discrete lineage of mother and daughter?
If I understand the question right, I think it's probably more common in unicells, as you suggested, although there are multicellular organisms that divide, that multiply by fission, like placozoans, for example, are actually animals that divide. They just divide in two like an amoeba.
Well, my question was, which the question of an animal that divides by binary fission is fascinating. But my question was, among unicellular organisms, is it more common for there to be the yeast model, or is it more common for there to be the E. coli model?
That's a great, very deep question. I think that the E. coli model is a little more common, but even that is not always as it seems, because there's been recent research that shows that even a lot of things that do binary fission, the way you're talking about, there is a sort of hidden asymmetry where, for example, one daughter, let's call it, one of the two products of division ends up with the older and more damaged proteins than the other one, and and if that keeps going, that lineage will eventually die of the accumulation of these damaged proteins. So there is an asymmetry there, even though you can't usually see it.
That's fascinating, and that speaks to like the depth of study that's required to be able to actually see these sorts of effects.
Yeah, I wanted to back it up a little bit and find out how you got into this question in the first place, and hear a little bit about your story of what got you interested in the question of multicellularity and how you came to be studying what you're doing today.
So I was doing a master's degree at the University of Central Florida, studying squirrels and the ways that squirrels are related to each other. And I took a course in mathematical modeling, and I decided to do a project, I don't remember why, on the evolution of multicellularity. We had to do a semester project.
Why were you into squirrels in the first place? What pulled you in that direction?
It was really sort of opportunistic. It was what one of my committee members was studying, and she needed to know the answer to a particular question about squirrels. And I had some of the skills to answer that question. So I decided to make that my master's thesis.
You were making yourself useful.
Yeah, trying to anyway.
Okay.
So in this project, I studied these guys, these algae called Volvox, which are a model system for the evolution of multicellularity. And I just got really interested in it. There's a book about Volvox written by a guy named David Kirk, who's kind of the father of the field. And so I read that book, and I was fascinated by the whole thing.
So what makes them unique? What is so fascinating about this particular organism?
Oh my goodness! Now we're
yeah.
So what's great about Volvox? There's two things. One is that for a multicellular organism, they're relatively simple. They only have two cell types. They have what we call somatic cells, which are the ones that die, that are mortal, and they mainly function for swimming. They each have two little hair-like flagella that they use to motivate themselves through the water. And then they have I don't know if your listeners are going to see this, but these bigger cells are what are called germ cells.
Yep, they can see. They can. Half of them can see, anyways. We release it both in audio and visual.
Okay, I wasn't sure about that.
But yeah, maybe we can describe it also for the listeners.
Yeah. So Volvox is a big green ball made up of lots of smaller green balls, and there's bigger and smaller of the small green balls. We're talking about the bigger ones right now. And those are gonidia that actually divide to make baby volvoxes. So, humans might have a couple of hundred different types of cells. You've got liver cells and cornea cells and skin cells, fat cells, and all that stuff. Volvox just has two. So,
Interesting.
partly because of that, it became a model for understanding the evolution of cellular differentiation.
And is this one of the only ones that we know of that has two, or is there a whole class of little
Wow, that is a great question.
little critters with two cells?
Two types of cells.
Sorry, two types of cells. Sorry.
I think it's pretty rare. I'm not going to say it's the only, because there's very little in biology.
And is the thought that they could sort of prove some missing link in this transition from unicellular to multicellular? Just to
I wouldn't say that, because they're not ancestral to anything.
I see.
But it's a model for understanding how cellular differentiation can happen.
Do you imagine that
Yeah.
Volvox could eventually develop a third type of cell? Like, is it something that you can push it towards?
That is something that we have talked about, and there's different opinions about that. I tend to think that probably it could happen. Other people think probably not, for reasons that might take too long to explain.
Well, it seems like if you were so from my limited understanding of types of tissues, you'd have to start getting basically, circulatory systems and organ systems, and it's hard to imagine how that could come just de novo from two cell types that are perfectly fine doing what it is that they're already doing.
Well, would it be the first step would be like towards a eukaryotic type thing? Is that what would happen? Like, there's organelles that appear first, or how does that leap happen? I mean, I guess these are individual cells. Are they eukaryotes? No. The algae?
Yes.
Yeah, they are eukaryotes, I see.
Not everything we call algae is eukaryote. Sometimes we call cyanobacteria blue-green algae. But I kind of lost the question.
Well, yeah, so hold on. Before we get onto the question of how you could push something that is two types of cells into more types of cells, you were sort of leading us through the arc of what it was that fascinated you about Volvox. So you're studying them because they offer you a model system for being able to understand what it looks like when you have just two types of tissues. And then where are the other things that sort of lead you down into the vibrant interest that is Volvox?
So yeah, that first aspect is really what David Kirk worked on for most of his career, was working out the molecular genetics of cellular differentiation in Volvox. What really fascinated me about Volvox is their relatives. So they belong to this group of algae where Volvox is actually the biggest and the most complicated in this group. There are actually single-celled algae in this group. There are algae with 4 cells, 4 identical cells, 8 cells, 16 cells, 32 cells, and so on, up to about 50,000 in Volvox. And that's a really powerful thing for using the comparative method in evolutionary biology, because you can look at that and you can say, well, what did their ancestors look like? What did the ancestors of any particular pair of algae look like? And if you have a big sample like that with lots of different, I want to be careful with this wording. I'm going to say intermediate stages, but I'm using that as a little bit of a metaphor because they're not really intermediate to anything.
Like there's no final form. And you're using that instead of saying species?
No, they are species.
Okay, they are species.
I'm trying to avoid implying that some of these simpler ones are ancestors of the more complicated ones, because they're all living species, right? The 4-celled ones are alive and happy today.
And they could just maybe be analogous for what an intermediary might look like or something like that.
That is exactly it, yes. Probably Volvox had a 4-celled ancestor and an 8-celled ancestor and a 16-celled ancestor. They're not the same things that are around today, but we can kind of use the ones that are around today as what, like you said, analogies for what Volvox's ancestors might have looked like. And so we can put together a sort of plausible series of steps through which a single-celled alga could have evolved into something like Volvox.
It's interesting there's no odd-numbered algae here.
Yeah, as a matter of fact, they're all powers of 2.
So that probably tells you something about how this happened.
Yep, it does.
There was some what would you say, like some sort of equal division going on, or something where somebody wasn't dying off immediately.
Well, it can't be like the budding yeast cell at the very least, right?
That's right. Yeah, it's actually critical to understanding their biology that their method of cell division is different from what we usually think about. So, in contrast to what we were talking about earlier, binary fission, where you start with one cell, it divides in 2, they divide into 4, they divide into 8, so on. These algae do something a little bit differently. They grow really, the individual cells will grow really, really big. So, for example, let's say you've got an 8-celled algae. Each of its cells will grow to 8 times its original size before it divides at all.
Hmm.
And then it will divide 3 times really fast, 2, 4, 8. It can happen in an hour.
And how long does it take to grow to the 8 times its normal size?
For most of them, it's a matter of days.
Interesting.
Maybe 2, 3, 4 days.
And so,
is this just a matter of scaling the size up, like getting the proper sort of cytoplasmic volume necessary to host the subsequent number of cells, or?
Yeah, it is. It's a matter of getting big enough, getting enough cytoplasm to divide into enough cells to make a baby. And that means that they basically start life with all their cells. So that 8-celled alga, the mother cell is going to divide 3 times to make 8 cells to make an 8-celled daughter colony. It's not going to divide anymore.
Like they have all of their materials in place, basically. It's just a matter of reshaping or something like this.
Right.
That's that's pretty unusual. So the structure that's behind you is an 8-celled structure that on the wall?
Well, this one's about 2,000 cells.
Okay, that makes more sense. But the ones that you study in the lab are much smaller than that one sometimes.
I study all of them.
Okay. So, okay. The Volvox is a model for studying the emergence of multicellularity because it has all of these characteristics that allow you to simplify the playing field, right? Because if you're trying to look at a human and to reduce the human to a functional model, that's almost impossible because there's just too much going on.
Yeah, and that's right. And also, we're so far diverged from our unicellular ancestors or our living unicellular relatives. Multicellularity probably evolved in our ancestors something like 600 to 800 million years ago. And all those intermediate species are lost. We can't look at them to see what our ancestors might have looked like that far back, unless we get really lucky with the fossil record.
Yeah, I was going to say, can a fossil preserve a single-celled organism, or you can see this level of resolution in the record?
You can, and there's lots of unicellular fossils. There have been some fascinating discoveries just in the last few years. But it's often, a lot of these single cells don't look that different from each other, even if they're very distantly related.
From the outside.
Right, exactly, which is all you can see in the fossil for the most part. You certainly can't sequence their DNA and see how it compares to other unicellular organisms. So it's really hard to know with certainty what a lot of these things were.
And is there an emergence of multicellularity happening right now that's sort of against the background noise of single-celled life on Earth that humans just aren't necessarily able to get their eye on? Or do you think that this is something that happened at a particular window of development of life on Earth that the window has closed and this is what we've got?
So I don't think it's the second of those two possibilities.
Okay.
Because when I talk about all these different origins of multicellularity throughout the history of life on Earth, they're not constrained to one particular time window. Volvox probably evolved multicellularity something like 200 million years ago. The brown algae, things like kelps, about the same time, about 200 million years ago. Animals, 600 to 800. Plants, maybe half a billion years ago. They're all different ages.
What's the
So
youngest
I don't.
version of that? That is the 200 million years the youngest? Yeah.
Oh, great question. Um,
Like, is it possible someone could go out into the woods tomorrow and scoop up a brand new species that's doing that has recently evolved to be, you know, 4 cells or something as opposed to the
And
original?
if they did,
Would you notice?
would
Yeah.
you notice,
Mm.
right?
yeah. So absolutely, that's possible. And one of the things that complicates this is how do we define multicellularity?
Mm.
There's actually a lot of gray area there.
I would think that it goes, 2, and then we're golden, no?
You would think, but there's actually a lot of organisms on Earth that have what we might call sort of marginally multicellular life cycles.
Hmm.
Because one thing you got to remember is that most multicellular organisms have a single celled stage in their life cycle, right? Even we do, right? The zygote stage is a single celled stage.
Mm.
That's true. I
Almost
remember these
everything has.
diagrams from biology, the ones that are the multiple cycles of like cell behavior. I remember always seeing those and just it causing a
Right.
headache.
Just for a second.
Yeah.
And there's lots of things that form sort of temporary multicellular structures. Um, Things like cellular slime molds, for example, live most of their lives as amoebae.
Like what is it? Discoideum?
Yep, yep. Dictyostelium, discoideum. Yep, that's the model organism in that
The
system.
slugs.
And that one is crazy, right? Because they live basically dispersed as amoebas, and then they hit it's a density dependent signal, and they aggregate in order to create this multicellular reproductive stock that then disperses
Seems
spores
like
that
it can kind of wiggle
stars.
around and act sort
Yeah, it
of
was
worm-like. And
we looked into this for an episode that we were doing on cheaters, and it seems like it's a model system for studying the emergence of cheaters because those that won't form the stock versus the fruiting body would necessarily crash the population, but it is a very strange
it's a
organism.
story of self-sacrifice and heroics and, the whole 9 yards. So.
Yep. Yeah, and it's a fascinating system, but I think you can begin to see there where there's a gray area. Like, would you call that a multicellular organism then?
Depends when you're
And there are
looking.
things that are even less kind of along that spectrum than that.
And so, and what does it look like to be less along that spectrum? So for me, I'm like, okay, so if it forms a body that moves around made of many cells that then behaves, I would say that that's well past the hard cutoff of multicellular. But
Okay.
where's the gray area? Where does the gray area live? Of well,
Yeah, how do
maybe.
you define multicellularity? I guess that's
Yeah, let's go there.
Well, let's start with your definition, which is great, because definitions are not right or wrong. They're useful or less useful. So we can pick one and we can run with it as long as we're explicit about it.
absolutely.
So when you're saying things like coming together to form a body, moving around in an organized manner, things like that, which I think is a fairly reasonable definition. There are I'm going to say quote single-celled organisms that sometimes in response to some kind of threat or environmental stress will form a multicellular clump that doesn't really do anything except sort of hunker down and wait for the poison to wash away or the predators to go away or whatever. And
So it would seem like
once
the
you've done,
intentional motion is critical then in the definition, perhaps.
I
The
wouldn't think
unicide
so.
motion, I suppose.
That wouldn't be part of my definition,
Okay.
but if we're going with that definition, then still there are gray areas here.
Mm.
There are things that form multicellular structures that do it temporarily, that don't have much coordination among the cells, that may break up again into single cells.
Are there times that that organisms form a multicellular structure that does not have a reproductive purpose?
Yeah, I would say there are
He
cases
just
like
mentioned like
that.
the defensive posturing and
The defensive, okay, all right. I guess if you zoom out far enough to the defensive posturing, I can see it being. If I've been that under reproductive, I think that reproductive begins to lose all meaning. So I'll avoid doing that.
The other really great example of gray areas, things like biofilms,
Mm.
which can be quite organized, but they can also be really simple. They can just be, so here's a good example. If you have if you grow E. coli bacteria on an agar plate, they'll form a little multicellular plaque on the plate. We don't think of that as multicellular. It is multiple cells living in contact with each other, but we don't think there's a lot of coordination going on there. We don't think that there's
like, does the division of labor play into the way that you define? Like, I guess this all comes down to what is an individual. What constitutes the individual, and is that necessary? Because like Dr. Benderbury during her PhD was looking at these biofilms actually, and as I understand it, there really was a great deal of complex. Specialized roles throughout the various depths of the biofilm in response to the different levels of oxygenation and things like this. So I don't know. I'm always very terrified by this idea that things are simple because it really seems to me that the closer you look at them, the more complex they actually appear to be. And
Yeah, I think you're right. But biofilms can be very complicated, can have division of labor, can have signaling going on between the different cells. I wouldn't say they always have those properties, but they certainly can. And there's an active debate about whether we should think of biofilms as multicellular.
And
But again, all this just depends on your definition.
I like that. Yeah, it's a very good point.
And to come back to where something that we touched on at the very beginning of the conversation. So what is the sort of the advantages versus disadvantages of becoming multicellular? Why would there even be such an abundance of unicellular life if you have the advantages of community goods and you have the advantage of division of labor and you have the advantage of size?
What are the top three theories? Like there must be people just arguing about this nonstop as to what actually drives this process. But do you have a top 10 like tick list? Maybe you can share a few with.
I actually do, because some friends of mine, some colleagues of mine, just published a preprint today that talks about exactly that question. What are the hypotheses for why multicellularity evolved?
Perfect.
Kai Tong is the lead author on that, and it's work out of Will Ratcliffe's lab at Georgia Tech. And so I can give you a few of them, but I really like, for a much more thorough discussion of that preprint is really a great resource, and they've got just a beautiful graphic.
Cool. We'll put it in the description, but
Okay,
maybe for
yeah.
the listeners, you can just briefly overview sort of
Or your favorites.
Yeah, what are your favorites? What's sort of the general line of thinking that's popular right now amongst the thinkers that are working on this?
So I think that protection from predation is a major hypothesis. And the idea there is basically just that you get to be too big to be eaten by whatever predator is common. We've actually tested this experimentally. We've done this with some single-celled relatives of Volvox, where we expose them to a predator, and they eventually, actually pretty quickly, in a matter of months, evolved multicellular structures that were genetically fixed in the population. So that's one, and I think there's good experimental support for that at least being plausible.
Too big to eat.
Yeah. Or at least too big to eat by the predators that are common in your environment. Another idea that is probably really relevant for things that don't move under their own power, like plants and algae. Well, a lot of algae. My algae swim, but lots of algae we think of as seaweeds, right? Kelps, things like green algae like sea lettuce, red algae like nori, the stuff your sushi comes wrapped in, um, which is, I think they call it, I think the Tong et al. Paper calls it competitive overgrowth, which is the idea that those organisms need a substrate to live on. They have to attach themselves to something. And there's immense competition for space on, say, a rock or a piece of coral or something like that. And so once you secure yourself a space, you want to capitalize on it as much as possible. So you get bigger so you can reproduce more. And at the same time, if you're a photosynthetic organism, you're shading out your competitors
Mm-hmm.
that are also on that rock or on that piece of coral or sand or whatever. And if you don't, they'll shade you out, and then you'll starve because you can't photosynthesize anymore.
So it's like an arms race of size and stability in the population.
Yeah, exactly.
And you can see that happening in a forest, just going out in the woods and looking around and seeing the little saplings getting squeezed out. And
yeah, we spend a lot of time in the Pacific Northwest. There's a lot of clear cuts, and so after you walk through the area, you can start to really identify what the forest looks like after year 1, year 2, year 10, and it seems like a microcosm for the sort of the evolution of behavior, because you can see that it becomes advantageous for one tree to grow as quickly as possible and be as tall as possible and take up as much sun as possible. And I imagine that there's immense warfare happening underneath the ground that's basically each of them attempting to prevent the other from being successful, but they've somehow managed to kind of balance that. But I imagine that that's the case in the bacterial and the single-celled world as well, where if you have someone who's basically beginning to accumulate a lot of resources, the competitors are going to try to nip that in the bud as much as humanly possible by producing as much toxic stuff as they can.
algae possible.
It's true, my mistake.
All right, so we have them out competing. We have this idea of making the most of space and of being bigger than that's trying to eat you. What else do you got?
Well, I want to just address one thing you brought up. That you're exactly right about this warfare going on all the time. And that's why we have antibiotics, right? That's exactly why fungi produce antibiotics to kill bacteria, because of this constant warfare. That's how penicillin was first discovered. So to answer the other question, let's see. I'm not going to remember all of them.
No, that's fine.
Some of them really apply more to aggregative multicellularity, which is what we were talking about with the dictyostelium.
Like a periodic multicellularity that's part of the life cycle.
Well, a lot of
all of us
clonal multicellular things are. I mean, we're kind of that too. But where they live a lot of their life cycle as single cells, and then they come. The difference between clonal and aggregative multicellularity. In aggregative multicellularity, you have these free living things that come together to form a body, and they're not necessarily all genetically identical to each other. All the cells that make up a dictyostelium slug or fruiting body. Whereas in clonal multicellularity, you generally start with a single cell, like we do, and it divides. So all the cells are basically genetically identical to each other.
So this is a really far out question, and I recognize that it seems almost impossible for the answer to be yes. However, so Lynn Margulis proposes the endosymbiont hypothesis, which is that mitochondria are an endosymbiont that is absorbed by an archaea that then becomes eventually the first eukaryotic cell. What about specialization? Is it possible that specialization, the way that you see in humans or more complex organisms than Volvox, comes from the coming together of multiple organisms that have kind of blended their DNA, and so everything is encoded, but now looks like a nucleus and an organelle. Well, so that's on the cellular level, but also looks like a heart and a liver and the intestines. Like, is that
That's a thing.
okay? All right.
That's totally a thing. If you look at lichens, lichens are a combination of a fungus. Actually, it's been very recently discovered that most lichens are a combination of two fungi and either an alga or a cyanobacterium.
That's
And
right, yeah. And it was discovered by someone who I remember came to science really late in life, which I think is a very inspiring story. Because he'd come as an adult to do grad school, and then he ended up in a laboratory where he was doing genetic sequencing, and he was the one who uncovered it after decades of people looking, which I thought was a very interesting story. I don't remember his name.
yeah, well, that that happened in the department that I was in at the University of Montana.
Fascinating. So were you there when it happened?
I think I was. I wasn't there when the paper came out, and I didn't know that it was going on. They
Sure.
didn't talk about it much until the paper came out in either Science or Nature. I forget which one.
But so you're using the example of lichen, which is these multiple organisms living in one body, multiple different lineages living in one body, to create another organism to be that it could be extrapolated outwards to something that's more intricate. Because do they
Specialized?
specialize? Like, do they have organs inside of a lichen? I mean, I've looked at a lot of lichen and they definitely look crazy, but I don't know if they necessarily have organ systems.
I don't know about organs. I would say that they definitely have tissues. At least some of the things like they call them fruticose lichens, things like reindeer moss, which is actually a lichen. Um, they're a pretty complicated structure, and I think you would say that they have tissues. I wouldn't necessarily go as far as to say organs.
Okay. We'll look into that. That's lichen's fascinating. We haven't done an episode on that, but I think that it's worth.
Yeah, I'm curious, like, to what extent this Well, I have two main questions. One is, where is all of this headed? Which I don't expect you to necessarily have an answer to, but it's very curious. Like, is it a teleological process? Is there an end point? Why is the Earth doing this? Why, if it's, obviously it's not outright evolutionarily necessary for all organisms to become multicellular. It seems like a particular little mission off to the side. And is it, are our societies, our cities, the next stage in that evolution, interaction between multiple organisms that eventually can't live without one another? And, where's all this headed? What's going on? What's the bigger process here?
So I don't know where it's headed. I don't think anybody does. I don't think that evolution is a teleological process. I don't think there is an end point, at least not one that's predetermined. I am a big believer in the role of contingency in evolution, which is something that that Stephen Jay Gould argued pretty fairly persuasively in favor of. His whole book, Wonderful Life, is largely about the role of contingency in evolution.
And can you sort of explain that concept to our listeners?
Probably not as well as he did, but
basically the idea that life has the ability to change its mind at the drop of a hat, something like that. I don't know. Am I on the?
Well, yeah, using mind metaphorically.
Yes.
Yes. The metaphor, actually, I can explain it pretty well by stealing his words.
Beautiful.
or stealing his ideas.
Paraphrase.
It's a borrow.
Yeah.
The metaphor that he used that is, I think, very powerful is the idea of rewinding life's tape. So he posited that if you could back up the so-called tape of life, if you could turn time back, say 500 million years to around the Cambrian, and let it play forward again, would you get the same outcome? And he argued, and it wasn't, he didn't just assert, he made some pretty, what I think are compelling arguments to support this, he thought that you would not get the same outcome, that if
Mm.
you ran the tape forward 1,000 times, you'd get 1,000 different outcomes. Now, that view is not universally accepted. There's a guy named Simon Conway Morris who very famously disagreed with Stephen Jay Gould about this. They had a series of exchanges in Natural History magazine.
Well, it seems like there's a lot of space for non-binary solutions to that question too. You know,
Yes.
things could, you might have very similar looking things, but you know, the odds, yeah. It seems intuitive to me that you wouldn't get the exact same outcome as well. And you see the people arguing this. This is a really hot topic just in the philosophical and metaphysical world, or even in physics for some strange reason, which is this determinism versus, you know, everything's up to you kind of situation. And
The determinism of it is actually interesting to me, and I've thought about this on the level of the limited shape relationships that are necessary in order to perform the functions that have been decided as universally necessary for all of life.
according to the environment, even.
According to the environment, right? Like if you have Earth-like conditions, Earth-like gravity, Earth-like chemical abundances, is it possible that all of the organisms that are viewed as being related to one another, or not even all of them, but some of them have just converged to the same genetic code, and they're actually, our understanding of everything fitting together on a single tree of life is just absolutely bananas wrong?
I think that is extremely unlikely.
Okay, all right, I'll take that. But I hear you say extremely unlikely, but not impossible.
Well, I'm reluctant to say almost anything is impossible, but here's why I don't think that's likely. I do think, okay, so convergent evolution is a hugely important process. I don't think anybody really disagrees about that. You know, there's so many examples of it that I could point to, and it does argue that, for example, something like a tree, like coming back to what you were talking about a while back, something like a tree is probably inevitable. And this is not my original argument. I think this goes back to Dawkins, but I'm not actually sure. The argument that if there is life elsewhere and it is photosynthetic, you know, the ecosystem or the biosphere is basically driven by sunlight or starlight, then something like trees probably exists, because there's always going to be competition for light. To get to the light, you have to get tall, just like you were talking about earlier. You have to crowd out your neighbors or overshadow your neighbors.
Mm-hmm.
And that imposes certain biophysical constraints. You have to have a certain amount of strength in your tissues to be able to grow tall. And there's going to be competition that will drive that. It's probably nearly inevitable on a planet that has liquid water that there's going to be something shaped like dolphins and swordfish and killer whales and what are they called? The ancient extinct reptiles that were
Ichthyosaurus?
Ichthyosaurs. Thank you. You know, that body shape has evolved many times because it's really efficient at going fast through the water. So we probably, if you rewound life's tape and played it again, you would probably get something that had that body shape. Now, it might not be a reptile. It might not be a fish. And there might not be, maybe there would be a mammal that was shaped that way, but maybe it wouldn't be a close relative of hippopotamus. Maybe it would be a close relative of squirrels, right? Maybe some other mammal lineage would have gone back to the water.
Swimming squirrels is terrifying, but I'll entertain it.
It is kind of, yeah. And there's flying squirrels, but swimming squirrels, that's a whole other ball game. And once again, I've lost the thread of the question.
The thread of the question was, is it possible for all of life to have shared genetic codes through this sort of mechanism of convergence? Because I hear you saying that, you know, this body plan evolves multiple times in order to do the same sort of function of moving quickly through water. And I'm like, well, then why is the, you know, F1, F0 ATPase or ATP synthase not also the best possible shape for making ATP?
Well, I don't know about that. I don't know why it's not the ideal shape for making ATP. But if you're talking about convergence at the DNA level, which I think is what you're asking, right? Is it
I guess
possible that,
honestly, this concept could go all the way back, right? You could really say, is life occurring even all of the time, and we're just missing it because it's so convergent of a process that it just looks the same every time, and we think
yeah,
it's on a tree when it's actually just occurring
yeah,
Well, because when
constantly.
you start to look at bacteria, you start to very quickly lose your ability to place them upon a tree. Where there's a lot of horizontal gene transfer, there's a lot of sort of when you start to try to reconstruct the last universal common ancestor, like Nick Lane has written about this, where if you start to do evolutionary reconstructions of a common ancestor, you very quickly realize that there's too much mixing and shifting between bacteria to be able to say so. So why is it not possible to have a convergent sort of production of gene sequences?
Or why is it unlikely? I
Yeah,
think
why is it unlikely to have a convergence of gene sequences that do the same sort of thing but not necessarily be related?
okay. So there's two questions
There are
there.
two questions. I'm
I
sorry.
hope I remember to get to both of them. The reason I think the second of those questions, the reason I think that's really unlikely is because there are probably multiple, well, we know there are multiple ways to achieve most ends. There are multiple gene sequences that will produce an enzyme to achieve a certain, to catalyze a certain reaction.
hmm
In fact, there are an immense number of DNA sequences that will produce an enzyme that to perform any particular catalytic function.
In other words, they
So,
could change quite a bit and still get the job done. Like you could have a million ways to make a house, and it would still be a house, essentially.
right. And what that tells me is that if I look at that enzyme in a human and I find the same enzyme in a, let's say, a choanoflagellate, the close unicellular relative of animals, I find that same enzyme and it has a very similar nucleotide sequence, then I'm pretty confident that the most recent common ancestor of those two organisms had that enzyme, that it didn't evolve convergently.
And these are like
Because,
exactly the same, right? You're talking about like this is
well, it doesn't have, it's almost never exactly
mm,
the
mm.
same,
Be-
but it might be 90% similar or
mm.
80%
Because you
similar.
do see like pyramids in Egypt and pyramids in the New World.
Well, not even that. You have butterflies that are identical in on one island that have absolute convergence in a different place because that's just it seems to make them
Butterflies and bats and
it just makes them more
both
fit.
have
And so it's
wings.
hard, this is something that I was always obsessed with. So when I was doing my PhD, we did a lot of sequencing, we were looking at the evolution of a protein. And the conventional idea is that you look at the protein, you look at the homology of the DNA sequence with other organisms, and then you reconstruct a phylogenetic relationship of where the gene that encodes the protein came from. And I always spent a lot of time wondering about the fidelity of that, because if you have something that's very, very similar and there's just a few nucleotides of difference, that's harder for me to imagine. But sometimes you have similarity that looks like it's basically chunks of proteins of different genes that have been sort of they've come together to create something that has some of the same pieces as one organism and some of the same pieces of another organism. And that to me seems more convergent-y. That's a very scientific way of saying that.
Yeah, and it is entirely possible that something like that could happen. But the suggestion that so I appreciate Nick Lane's argument, and I don't disagree with it at all. But you're talking about a very, very deep part of the tree of life, right?
Absolutely.
Whereas if we look a little bit more shallow, and we look at a particular enzyme in animals, I can predict with a high degree of certainty that the human version of that enzyme is going to be more similar to the chimpanzee version of that enzyme than it is to the squirrel version of that enzyme, and it'll be closer to the squirrel version of the enzyme than it will to the earthworm version of the enzyme, and closer to the earthworm version of the enzyme than it will to the choanoflagellate version of the enzyme. Right?
But
There's an order to these things, and I can predict with a high degree of certainty that everything I just said will be true of most enzymes.
Will
but
all
they all have similar jobs too.
They do have so yes, they do all have
Like
similar
the
jobs.
monkey and the human have similar physiological requirements.
But my question is that does that apply to all enzymes? Like if you were to look at all of the enzymes that are in the squirrel versus all of the enzymes that are in the human, would they all be more similar to one another than they are between the human and the choanoflagellate? Or is there some randomness there that emerges as you're looking at similarity? And I don't know that anybody really, I've never seen this sort of analysis. I think that because it's very difficult, I think a lot of genes are not annotated. I think that we have these gross measurements of, this is the percent similarity between human and chimpanzee or human and squirrel, but I'm not sure if somebody has gone through and actually looked to see that, okay, across the board, the similarity between these enzymes is very, very, very high versus there being randomness in that.
Yeah, I mean, I can't swear that there are no exceptions. There could be cases where, for example, one particular gene has a very high mutation rate in one particular lineage. Let's say in the squirrel lineage, this gene that codes the enzyme has a high mutation rate for whatever reason, for reasons I don't understand. Then it could be more different from the human version. But if you look at all 20,000 or so enzymes, you're going to find that I would be very confident that 19,900 and some of them are going to fit this pattern, at least at the gross level I've talked about, where I'm only looking at four taxa or 5 taxa, something like that. For the other question, I don't want to forget this one because it is a really interesting question. How do we know life isn't constantly arising? We don't know that it isn't. We don't know for sure that life isn't constantly arising. But what we do know with a really high degree of confidence is that everything alive on Earth today that we've ever described, that we've ever discovered and sequenced its DNA descends from a common ancestor. And the reason I say that basically goes down to the genetic code, because there's nothing that we know of that says that you couldn't have a different genetic code. And well, in fact, we have some small variants of the genetic code, but everything on Earth basically has the same translation table from DNA to proteins. And there's no reason that has to be that translation table. There are an immense number of possible translation tables, an immense number of possible genetic codes. So this, in my mind, is actually how if we ever do discover life somewhere else, this is how we'll know whether pangenesis is true, whether if we discover life on Mars.
Yeah, I was going to say, would that change your hypothesis if you start looking out into the greater galaxy and you start finding that everybody is using this template and using the same DNA? Would it change your mind that perhaps this could be, well, obviously at that point it would either be convergent or you're looking at some super widespread panspermia type thing?
Right. And I would actually, if we found out that everything in the universe had the same genetic code, I would be more likely to believe in panspermia than convergence as an explanation.
Why is that? Just because you think there's so many possibilities. Why this one?
Yeah, exactly. So if we found life on Mars and it had the same genetic code or a close variant to what we find on Earth, I'd be pretty convinced right then and there that either Earth seeded Mars or Mars seeded Earth.
Mm-hmm.
That would be the first thing I'd want to know is what's its genetic code.
Mm-hmm.
Well, if we're not careful, it sounds like we are going to seed Mars. So we met with one of these actually a biologist who was working. Was he a biologist? He was working on one of this Perseverance mission, and he was very, very concerned that we were going to be taking little pieces of our home over there. So it seems like it's almost would be a remarkable feat of luck if we didn't seed Mars with something. Whether it can take roots and make little Mars cities on its own or anything like that has yet to be decided.
Yeah, because it's almost impossible to truly sterilize a spacecraft.
Exactly.
So then, where do you fall on panspermia now? Right, because evolution of multicellularity is pretty far upstream from evolution of life. But I assume that
Yes.
I assume that you have an opinion.
That's all it is an opinion. I think it's more likely that life evolved on Earth than was seeded from somewhere else. But, I don't feel super confident about that opinion. I wouldn't bet my life on it.
So, and this is just because it's too unlikely for something to have arrived fully formed on Earth, or is it just sort of it feels right, or where does that come from?
Or you just think that the mechanism for its independent origins is plausible enough that why not?
Well, because it's one of those things we talk about this a lot because okay, so life has, and I know that we didn't tell you that we would ask all of these questions before we started, so I'm sorry.
Right.
But we talk about this a lot in terms of, is there a plausible mechanism for the emergence of life if we cannot recreate it in a laboratory?
Or find it happening in the ocean.
Or find it happening in the ocean.
Well, I wouldn't expect it to happen in the ocean.
Interesting.
Because the ocean's not the same as it used to be, or
the ocean's not the same as it used to be, and there's so much competition for nutrients that anything that was on its way down that path and had the potential to evolve life would just get eaten.
That has always been my rationale for it as well, because I'm like, how could you possibly populate a place that's already populated?
As far as, do I think it's plausible, even though we can't reproduce it in the lab yet? Yes, I do think it's still plausible. I don't think that because we don't know how something happened, we can say that it didn't happen. So I do think it's plausible. I also think that there are limits to what we can know. So we will probably never know how life evolved on Earth. I think the best we can hope for is sort of like I talk about with multicellularity, is to identify plausible routes. I don't think we're going to be able to say this is how it happened.
Of course, yeah.
I
think so much could be said for all of science in general. That's something we sort of come across over and over again, is that we're really just restricting this possibility space.
Right, but in this case, we're not even restricting it, because it could be, that 10 years from now, somebody gets the Nobel Prize because they managed to evolve life in the lab from completely, inarguably non-living substances.
I guess the possibility space would be what mechanisms could possibly achieve that end.
Right. So that still won't rule out that it happened a different way, a way that we never conceived.
Right on.
And so and this is one of those questions that you don't ask a scientist, but I'm going to ask it anyways. If you can't come up with the answer, what is the point of the exercise of searching for it? Like, why does science look for the answers of the origin of multicellularity or the origin of life, or you can go all the way out to like the Big Bang. This is
curiosity.
Yeah, it is curiosity, but curiosity it's more satisfying when directed at something that you can point to and say, we have figured this out. And so, like, what is it that drives you and that continuously gets you interested in this question that you still, after studying it for many years, are still captivated by it?
There's a lot to unpack there.
Well, I mean, because this is a question that on some level you have recounted that you will never be able to answer. And so what is it that drives someone who is occupied with an unanswerable question?
Okay, so for me, I am a big advocate and fan of curiosity-driven science. I want to know how things work. I want to know how things have worked and how they can work. I accept that there are limits to what I can know. And that's unfortunate, I think. To me, it is. It seems that way anyway. But this is the best I can do. And I may not have always appreciated that. It's something. This is a question of epistemology, right? What can we know? And I'm arguing that for a lot of things in evolution, the best we're ever going to be able to do is say what's plausible, or to identify some things that are plausible, not to say this is how it happened. I don't think I always appreciated that, as a starting grad student, when I was, full of fire and convinced that I could do anything because I had sequenced DNA.
Well, there's a lot of gray space again between we can't know anything and we can know everything. And people tend to get hung up on these extreme possibilities, I feel like. And it often holds back a lot of discussion because we have a tendency to want to be extreme about all of our ideas about how to go about things.
Yeah. I'll be honest, a part of it too for me in the particular field that I'm in, and we've touched on several things that touch on this, is the prevalence of creationism in this country, in the US. And so much of that boils down to these sort of so-called God of the gaps arguments, that because we don't know how it happened, it can't have happened, right? Because we don't know how life originated, life cannot have originated through natural processes. Because they don't say this so much anymore. They used to, but it used to be because we don't know how multicellularity evolved. It can't have evolved, at least not through natural processes. And so by showing that there are plausible paths to multicellularity, hopefully eventually we'll find a plausible path to life. You break down those arguments.
You sort of take the mysticism out of it to some extent. You at least offer a rational alternative to the mysticism should someone be interested in pursuing that.
Yeah, and Darwin was the first one to do this. He has this example of the vertebrate eye. So he foresaw this argument, and he made what to me is a pretty convincing argument against it. In 1859, he showed that, in fact, there are lots of plausible intermediates between just a patch of pigment cells and a vertebrate eye. And we know that they're plausible because they exist in living organisms. Now, this is the caution I was making earlier. Those living organisms are not ancestors of vertebrates. So it doesn't show that is how it happened, but it shows that it's plausible. It shows that it could have happened that way. Mm-hmm. And very similarly with these algae, that was one of the things that I tried to do in grad school, was to describe a plausible series of steps. Actually, I should give David Kirk credit for this. David Kirk was the first one to describe these steps through which a single cell could evolve into something like Volvox. What I did is try to place those steps in order, basically.
And is that what you're still working on in the lab today, or?
To be honest, that is a lot of what my grad student is, or he's now my former grad student. He has graduated. That is a lot of what he's working on. And he may show that a lot of the stuff that I found was wrong. He already has shown that some of the stuff that I found was wrong.
Such is science, a real work in progress.
But you were gonna, you had a concluding thought. It sounded like you were saying, to be honest, to me, because you were kind of wrapping up this idea of why study this question.
Well, that is a motivator for me, to basically break down these creationist arguments that are, you can call them gods of the gaps arguments. You can call them arguments from ignorance. I don't know how it happened, so it can't have happened. Nobody knows how it happened, so it can't have happened, is a more fair way to describe those arguments. So yeah, to me, that is a motivating factor.
And I mean, I think that that's a very powerful motivator, and that's something that we spend a lot of time thinking about too, where it's, science is a field of possibility, and it seems that there are many questions that cannot be answered, and the joy is to find the things that are possible, or the things that are captivating, or the things that are interesting. It's this way of looking out onto the world in a more mechanistic way than perhaps most people are used to, but it does seem to order the universe in a way that is very satisfying.
And in its best form, could perhaps even lend itself towards bettering the world around us in terms of technological applications of the knowledge and so forth.
Well, that's the thing, isn't it? Right? This is this is one of the big reasons I'm such an advocate for curiosity-driven science, or what you might call pure science, is that um, you don't know ahead of time what scientific discoveries are going to translate into things like you're talking about, things that actually have direct benefits for human beings. You can't know that ahead of time. If you try to funnel your research into things that you're confident are going to have those kinds of outcomes in the short term, especially, then you're not allowing for the possibility of discoveries that you haven't anticipated, or applications that you haven't anticipated of discoveries that you might not otherwise have bothered to study.
Yes, yeah. And
Definitely.
you see this come up a lot. You hear, especially basic biologists, like complaining when they have to fill out these grants that they're always having to sort of chisel them into something that has a direct application, when really we should be valuing the fundamental knowledge itself because of what might grow up from that decades, from now, or what may come.
Do you have a sense for what can be done to prevent that from being a problem as someone who studies very basic research?
I'm actually pretty reluctant to talk too much about that for a very specific reason, which is that I'm currently doing a rotation as a program officer at the National Science Foundation,
Mm,
and I don't want to do or say anything that could be interpreted as representing
of
NSF.
course. Perhaps,
In fact, I could get in big trouble if I did
Let's
that.
not get you in trouble.
yeah, let's not get, but it would be fascinating, when you're done with the rotation, if it's something that you feel like you could comment on as someone who has had the direct experience with overseeing something like that, to be able to give some feedback, because I think that this is a huge, I've worked in basic research labs my entire career, and this has always been a concern that's on the back burner, where people are very.
And I, on the other hand, have always worked in technological engineering labs my whole career, and so we've had very different experiences when it comes to, paying the bills.
I'll bet.
And even like the kind of science that comes out of those labs, and like how people define science, and it's an entire world, but we won't get you in trouble.
Okay, thank you.
I think that we've taken a lot of your time. There's obviously other questions that have remained unasked and unanswered. I suddenly had the thought of asking about viruses, but I think that that's tangential enough that we will have to leave it for another time.
Okay, well, I've had a lot of fun talking with you.
It's been our pleasure.
You have a book coming out, as I understand it. Is that true?
Oh yeah, not just me. Along with Will Ratcliffe and Peter Conlon, we are the editors of a book that's going to be coming out in March. Actually, we have a publication date now. It's called The Evolution of Multicellularity, and it's about the evolution of multicellularity. We didn't
Cool.
want to make a mystery out of it.
Yeah. Well, everybody, go out and
Uh,
check this one out when it comes out. Where's it going to be? Is it going to be everywhere? It's going to hit the bookstores worldwide and straight
Great
to the New York
question.
Times bestseller list.
It is already for sale on Amazon, even though it's not out
Oh,
yet.
cool.
And actually, but you can get a better deal if you go to, I think it's CRC Press. I can provide the link to
Sure.
that. It happens to be the pre-orders are, I think, 20% off for a period of time that I'm not privy to.
Cool. I want to check
They
it. I want
call
to
it
read it.
November sale, so maybe it's November. I don't know.
All right. Well,
Uh,
we'll put a link in the description so people can check it out.
Other than that, is
And I should say,
oh
sorry,
yeah,
I should say it's an edited volume. We didn't write most of it. We have a pretty large number of authors who each wrote one chapter.
so it's like a collection of essays, or how would you describe it?
Yeah, well, the way I would describe it, I think best is as a collection of review articles.
Nice.
So we gave each author or set of authors um, guidelines on what we would like them to write, which was mainly to synthesize one particular aspect of the evolution of multicellularity.
Excellent.
Brilliant. Looking forward to it.
Mm-hmm.
Other than that, is there anywhere that folks can find you? Do you do social media? Is your lab website the best place to catch up on what's going on in your life lately, or going on in your lab life?
Yeah, I'm X Princeps. It's X as in X-ray on Twitter. My lab website is matthewherron.net, I think. matthewherron.something. I don't
Google it, folks.
.net.
DuckDuckGo it, whatever you do.
Yeah, it's not .gov for sure.
Stay away from his government involvements. It's not that.
And that's pretty much, most things of import I post to Twitter. I do have a blog that I have not been very active on lately. It's called Fierce Roller because that's what Volvox translates into, Fierce Roller, where I've written about all kinds of different things. But to be honest, I'm not publishing a whole lot there right now.
All right.
Fair enough.
We'll keep an eye in the future.
All right. Well, thanks for having me. I really enjoyed it.
Thank you
Dr.
for
Herron.
joining us.
Bye.
See ya.
Bye-bye.